The purpose of our project was to distinguish two children and declare their biological parents as they were switched at birth. In order to do this, we took blood samples from both Mr. and Mrs. Jones, as well as Mr. and Mrs. Smith, along with both children. Our hypothesis was decided by random as we had no indicators to help guide us. Our hypothesis is that child 1 would belong to the Jones family and that child 2 would belong to the Smith family. As for the test itself, our materials included 12 toothpicks, 6 stimulated blood samples, 1 stimulated anti-A serum, and 1 stimulated anti-B serum, as well as 6 blood typing trays. Now, blood type is determined by the presence or absence of certain agglutinogens that are located on the surface of erythrocytes. If there is a presence, a cloudy and foggy mixture will appear. This is called agglutination. Within agglutination are antibodies. This is important to know for the purpose of blood transfusions. People can only receive certain blood types. For example, if a person with the blood type B is transfused with blood type A, the recipient's anti-A antibodies will attack the type A erythrocytes. In simpler terms, antigens have been designated as A and B. A person with A antigens has anti-B bodies. To determine each individual's blood type, we sorted each plate into 2 parts, using the same blood except one is labeled A and one is labeled B. This is because we will be inserting 2 different anti-serums to check for agglutination. If agglutination only occurs in the place that the anti-A serum was added, the blood type is A. If agglutination occurs only in the place that the anti-B serum was added, the blood type is B. If agglutination occurs in both, this indicates the blood type is AB, and if there is no agglutination in either, the blood type is O. Our results indicated for child 1 that agglutination appeared in B. This made it clear that child 1 belonged to Mr. and Mrs. Smith, as Mr. Smith's blood type came back as AB, meaning Mr. Smith was able to pass down that B to his child. If Mr. Smith had blood type A, then this would not be a match, as Mrs. Smith also had blood type A. For child 2, no agglutination occurred, meaning he had type O. This matched Mrs. Jones' blood type, meaning child 2 belonged to the Jones family.
Ward's World produced the science kit used in our experiment, and the following chemicals were used: styrene C, lead, and lead compound Cr. Styrene C is moderately toxic to aquatic organisms and has a low toxicity to land animals. The Land Protection Agency lists styrene C as a hazardous air pollutant, and the Government of Canada even lists the environmental effect of styrene as unknown, but the amount in Canada is not harmful to human health. Lead and lead compound Cr are detrimental to our environment as they contribute to the loss of biodiversity and decrease growth and reproduction rates in both animals and plants.
Sometime around the 1900s, Carl Landsteiner discovered there were 4 different kinds of blood that were determined by the presence or absence of specific antigens on the surface of red blood cells, which is also referred to as erythrocytes. Antibodies against A or B antigens begin to build up in the plasma shortly after birth and decline slowly throughout one's lifespan. There's no clear stimulus for antibody production. However, it has been proposed that is initiated by amounts of A and B antigens that may enter the body through food, bacteria, and other means. So humans produce antibodies against those antigens that are not on the red blood cells. Just like we learned before, a person with A antigens has anti-B antibodies, and vice versa. A person with neither A or B antigens has both anti-A and anti-B antibodies, and a person with both A and B antigens has neither anti-A nor anti-B antibodies. Human blood types are inherited by multiple alleles. This occurs when 3 or more genes occupy a single locus on a chromosome. These blood groups and other inherited antigen characteristics of red blood cells are often used in medical legal situations involving identifying paternity cases and disputes where babies may have been mixed up. However, blood typing cannot prove if a man is a father, but it can exclude an alleged father. Blood typing has profound effects on society, influencing various aspects such as healthcare, ethics, and policies. For healthcare patient treatment, knowing blood types is crucial for safe blood transfusions and organ transplants, preventing adverse reactions like injury to the lung and destruction of red blood cells. On the other hand, blood type compatibility is assessed during pregnancy to prevent Rh incompatibility issues that could harm the fetus. When looking at ethical and moral issues, informed consent comes into play. Issues may arise regarding the collection and use of genetic information associated with blood typing, raising concerns about privacy. Efficient blood typing contributes to cost-effective healthcare by reducing complications from transfusions and ensuring proper allocation of resource. The advancement of blood typing technologies involves investments in research and development, influencing economic factors. Something important to note is that the government may need to establish and enforce regulations regarding the collection, storage, and use of blood type information in order to protect individuals and maintain public trust.